Good afternoon, everyone, and welcome to our Thursday afternoon webinar session. We are excited that you are joining us today. I'm Melinda Bikini, Director of Patient Services with the Cholangiocarcinoma Foundation. I'm also a patient, a 13-year survivor of stage 4 intrahepatic cholangiocarcinoma. And today I'm excited to say we are joined by Dr. Caitlin Van Lith, who is a fourth-year MD PhD student at the Mayo Clinic doing her PhD research on the genetics of cholangiocarcinoma with Dr. Lewis Roberts. She hopes to pursue a career in emergency medicine and global public health. And today she is going to be talking to us about we call it the GWAS study, the global study of the genetics of cholangiocarcinoma. And Caitlin, I will turn it over to you. Thank you for joining us.
Great, thank you, Melinda. So like Melinda shared, my name is Caitlin. I'm a fourth-year MD PhD student at Mayo. The MD PhD program is 2 years of medical school, 4 years of PhD work, and then another 2 years of medical school. So at the end of this, I'll be a doctor. And I'm doing my research with Dr. Lewis Roberts, who is an expert in hepatobiliary cancers. He stands out in the field of cholangiocarcinoma, and he is spearheading this project that is a global study of the genetics of cholangiocarcinoma. So let's see, there we go. So the objectives today are, we're going to start out by reviewing cholangiocarcinoma. I know a lot of you are experts on this in your own right, but just going over the basics of it. And then talking a little bit about what we know about the genetics of cholangiocarcinoma. And then we're going to talk about what a GWAS is, a genome-wide association study. And finally, we're going to talk about the International Consortium for the Genetics of Biliary Tract Cancers, which is really what this project is about. There's a discovery phase that's already happened, and now I'm working on the expansion phase as part of my PhD research. So starting out with reviewing cholangiocarcinoma, this is a cancer of the bile ducts, and it can be intrahepatic or extrahepatic. Here you see a schematic of the liver. This is the gallbladder, and all of these green branches here are your bile ducts. There are a lot inside the liver, and then it goes down through your pancreas into your small intestine, where it drains out through your colon. So intrahepatic cholangiocarcinoma is going to be a tumor that's within the liver. You can have perihilar cholangiocarcinoma. So this part of the liver right here is called the hilum. So perihilar cholangiocarcinoma is right there in the hilum. And then you can also have it on the bile duct that goes to the small intestine, and that's called distal, or this is part of the extrahepatic cholangiocarcinoma. So there are some well-recognized risk factors of cholangiocarcinoma, including colodocal cysts. So this is a congenital cyst. That's part of your bile ducts. It's pretty rare. The population that has it the most is Asian females. My little sister actually had a colodocal cyst that she got repaired when she was 2 years old. Another common risk factor is biliary tract stones, gallstones, primary sclerosing cholangitis. This is a big risk factor. This is sclerosis or scarring of the bile ducts. And then liver fluke infection, which is a parasite that is most prevalent in Southeastern Asia, especially Thailand. So this is less of a risk factor in the United States, but globally, it's important to keep in mind. However, most cases of cholangiocarcinoma don't have a known cause. So while these are risk factors, not every patient and not even a majority of patients who are diagnosed have any of these risk factors. And because of this, early diagnosis is difficult because there's not necessarily a group of patients that is being monitored for cholangiocarcinoma in the way that breast cancer or liver cancer groups of patients are able to be identified to monitor, but cholangiocarcinoma is difficult to monitor for because of the lack of known risk factors. And because of this, prevention and screening are really key to the diagnosis. So this is a map of the cholangiocarcinoma mortality rates throughout the world. So this is a reflection both of care in the countries and then also prevalence in the countries. And the really important thing that I want you to note as part of this is how much of the world isn't represented. So there's a lot of data in North America, some in South America, and a lot in Western Europe. But a majority of Asia, every country in Africa is not even being represented on this map. So this isn't because cholangiocarcinoma doesn't exist there, but it's because the research in those countries lags behind a lot of the world in so many ways, both because of lack of resources and then also because of lack of attention from countries that do have resources. So I want you to keep this in mind as we go through the presentation, because this is really important to the project that we're undertaking. So like we talked about, there are a number of known risk factors for cholangiocarcinoma. None of these, again, are all-encompassing, but they include inflammation, liver fluke infection, and then on the right there, there's primary sclerosing cholangitis. And it's important to note how these arrows kind of all interplay with each other. And so we're going to be looking at host genetics, and host genetics influences both risk factors and the development of cholangiocarcinoma itself. So now we're going to review the genetics of cholangiocarcinoma. And the one-liner for the genetics of cholangiocarcinoma is that it's complicated. There are a lot of cancers where there are strong known genetic risk factors. If you think about BRCA mutations in breast cancer, that's something that is kind of well known in the public for being a risk cancer for breast cancer, and some other cancers, ovarian and prostate. However, there's no gene like that for cholangiocarcinoma. The links in families are not obvious. There's low heritability in families for cholangiocarcinoma itself. And most of the genetic links that we can find in cholangiocarcinoma can be attributed to genetics of the risk factors. So looking at the genetics of primary sclerosing cholangitis, predisposition to gallbladder issues, those are the things where there is an obvious genetic link, but for cholangiocarcinoma itself, there's not a lot of research and there hasn't been any obvious findings thus far. And then also, when looking at mutations in cholangiocarcinoma tumors, a lot of them are common cancer-causing mutations in general. So these aren't specific to cholangiocarcinoma. A lot of the ones that we see are seen in many types of cancers, kind of some of them in like every type of cancer. And we'll look at that a little bit in the next couple of slides. And another thing that makes cholangiocarcinoma genetics complicated is that the genetic profile is different between intra and extrahepatic cholangiocarcinoma. It's not a cancer that is homogeneous or the same between the different types of tumors. And that's important to keep in mind. In a way, intra and extrahepatic cholangiocarcinoma are almost very different diagnoses. The treatment might be different and the disease-causing mechanisms itself are different. And when we're looking at the genetic studies that have been taken so far in cholangiocarcinoma, a lot of them have had, sorry, the mailman just came. One second. Every single day. But a lot of the genetic studies that have happened in cholangiocarcinoma have been in very small numbers. Cholangiocarcinoma is a relatively rare cancer, and so it's hard for an individual research center to see enough patients to have large numbers of cholangiocarcinoma patients in their studies. And because of this, it's hard to get those like really large groups of patients that are important to doing genetic studies, especially. So I talked a little bit about mutations that are common in a lot of different types of cancers. So in this graph, the blue lines or the blue bars are cholangiocarcinoma tumor types. And you can see along the bottom, these are different proteins or genes that have mutations. And the specific identity of these proteins isn't important to the scope of this presentation. But I want you to know that IDH mutations, KRAS mutations, TP53 mutations, BRAF mutations are common in almost every tumor type. So this isn't specific to cholangiocarcinoma. For example, TP53, P53 is a protein that's involved in DNA repair. And so cancers occur when the DNA is mutated to a point where it doesn't stop the cells from replicating. And P53 is an important protein to stop that. So almost everyone with P53 mutations will eventually develop different cancer types. And so the fact that this is coming up in cholangiocarcinoma is very unsurprising and is completely non-specific. And then looking again, this is separated by intrahepatic cholangiocarcinoma and extrahepatic. And you can see that the tumor profiles of these two types are different as well. So this is the things that I want you to get out of these graphs is that the genetics of cholangiocarcinoma, what we know so far is pretty unspecific to CCA. And then also it's hard to differentiate between intrahepatic and extrahepatic cholangiocarcinoma when you're just looking at cholangiocarcinoma patients as an entire group. And because of what I talked about each center having a very small amount of patients, that makes it even harder to divide the patients up between intrahepatic and extrahepatic, because that small number in the study then gets divided further. And so a lot of these differences are just really hard so far to suss out. So now we're going to talk about what genome-wide association studies are and how they can contribute to cholangiocarcinoma research. And in order to start that, we're going to just do a brief review of genetics. So this over here is a chromosome, and you have 23 pairs of these. And that's why the company 23andMe is called what it is. This is the double helix that you learn about in high school. And then the DNA is made up of 4 different types of nucleotides, is what these are called. But the way that we name them is A, C, T, and G. And they pair up in certain ways. All the A's and T's pair, all the G's and C's pair. But what's important out of this is that you have 4 different base pairs that make up your entire genome. And these are really important. They're just like the basic building blocks of who we are as people, essentially. And then in your whole genome, you have parts that are a gene. So in this example, I picked an arbitrary length and labeled this as a gene. And then you also have parts of your DNA that aren't genes. They're running in between genes. So maybe this is a gene, and then this isn't, and then maybe another gene starts here. And that's really important to keep in mind when you're talking about the genetics of disease, looking at specific genetic variations and whether they occur in a gene or outside of a gene. And so we're going to be talking about single nucleotide polymorphisms, or SNPs. So that occurs when one of these base pairs, so this was originally a C, gets changed into a different base. So instead of a C, it's an A, and then A's pair with T's. So this one, the G, is going to turn into a T. So there are a lot of different reasons why this happens, but this is a schematic of what that looks like. So when we're talking about these single nucleotide polymorphisms, or SNPs, these are the most common type of genetic variant. This is the kind of variation that you would hear as a mutation. The word mutation isn't encouraged in science just because that has such a negative connotation, and a lot of these SNPs aren't disease causing. So we call them genetic variants because they occur very normally. They're one in about 1,000 nucleotides, or those ACGT bases. 1 in 1,000 contains one of these SNPs, which means that each individual has 4 to 5 million SNPs in their genome, and you don't have 4 to 5 million diseases. So a vast majority of them are non-disease causing, and kind of part of that is because a vast majority of them don't occur within genes. They occur in those spaces between genes, which are a little bit less important. So if, though, there is a genetic variant within a gene, so genes create proteins, and if there's a variant within a gene, it can impact the function of that protein. So when I talked about P53 being a common cancer-causing mutation, I said that that protein is really important in stopping abundant replication. So tumorigenesis, making tumors, P53 is really important in stopping that. So if that gene is mutated and the protein doesn't work, then we don't have anything stopping it. And so any mutation, especially with, there you go, I used mutation, any genetic variant within a gene might predispose an individual to developing cancer. So genome-wide association studies, what we call GWAS, it's sequencing DNA to look for these SNPs. So the important thing that separates GWAS from some of these smaller studies that I was talking about is that it is sequencing large numbers of patients. So in our case, we're sequencing large numbers of cholangiocarcinoma patients, and these are our cases. Then we're also sequencing large numbers of people without cholangiocarcinoma diagnosis, and these are our controls. And so with these large numbers, it allows for identification of rare variants that are associated with CCA. And as I said, these can be much less obvious in small populations just because they are rare. And so when you're looking at large numbers of patients, it just becomes those rare variants you might start seeing more than 0 or 1 or 2 times. And so they might become more obvious in these studies. So this is a schematic of a GWAS study. Up here you have the gene with the ATCG that we talked about. And this red variant, this T, that's one of those single nucleotide polymorphisms that we talked about. And this group of patients right here are the cases with the disease. Let's say all the red ones have this SNP. These are unaffected controls, people without the disease, and let's say all the red ones have this SNP. And so if we're sequencing small amounts of people, like just this bottom row, you're like, okay, 3 of these patients have this SNP and 5 don't. And this one, 2 of the controls have the SNP and 7 don't. And you're like, the difference between those isn't super big. So we can't necessarily associate that SNP with the disease. But if we're sequencing large numbers of patients, we're suddenly seeing a difference between people with the disease and people without the disease. And the way that we analyze this data is through this plot down here. This is called a Manhattan plot because it looks like the Manhattan skyline, especially when you have a lot of hits and you have a lot of these lines coming up. So each one of these dots is representing a statistical test between the affected cases and the uninfected controls at that point in the genome. So the x-axis here is those 23 chromosomes. It's often 22 chromosomes because the 23rd one is the sex chromosome, so either X or Y. So you can see like that all of this light blue is on chromosome 1, 2, 3, and these are all testing for individual SNPs. So there are a ton of data points in this graph. But the higher up it is on the y-axis, the more of a difference we see between the cases and the controls. So for example, this red variant that this example talks about is coming up here, and you can see it's pretty high up. So there's a decent chance that this is actually more common in the cases than the controls, which we wouldn't have been able to see without large numbers of people. And so now we're going to talk about the International Consortium for the Genetics of Biliary Tract Cancers and why this study is novel and important in the field. So this is an effort led by my boss, Dr. Lewis Roberts at the Mayo Clinic, and Dr. Catherine McGlynn, who works at the National Cancer Institute. And this is the first GWAS of cholangiocarcinoma. Like I said, genetic studies of CCA thus far have been in small numbers of patients, but our goal is to get a lot of patients for this study. So between 2016 and 2019, Dr. Roberts and Dr. McGlynn led a discovery phase. So they recruited 2,610 cholangiocarcinoma patients from 24 different sites. And all of these sites are in North America and Europe. So this is the distribution between, and Australia. This is the distribution between the different countries. And we got, again, this 2,600 number of patients with cholangiocarcinoma. And so when you're thinking about discovery phases of scientific studies, these are used as sort of a proof of concept. So had this discovery phase not turned up a lot of things, then it wouldn't have make sense to go on to an expansion phase. So we're going to look a little bit at what the discovery phase found. This is, again, a distribution by male and female, intrahepatic and extrahepatic, whether or not the patients had primary sclerosing cholangitis, and then non-Hispanic or Hispanic ethnicity. And so this is, again, breaking it down into smaller numbers. Like I was talking about, the smaller the number, the harder it is to see differences. So even in this group, 2,600 sounds like a large number, but as you break it down, you keep getting smaller and smaller numbers. So this is an example of the Manhattan plot for all of the samples from patients that were of European descent. So this is 2,400 different patient samples. And again, the higher up it is on the y-axis, the more significant it is, the bigger difference there is between cases and controls. And there are two lines that we kind of look at these graphs. One is a little bit lower, and this is the line where it's standing out from the bulk of the data. You can tell that this dot is higher up than the rest of the dots in that chromosome. But when we run statistics on that dot, it's not coming up above the line of statistical significance, which is this red one up here. So any of the SNPs, single nucleotide polymorphisms, that are above this red line are ones that are coming up statistically significant. And the thing with statistical significance is it's easier to see smaller differences in bigger groups of people. So the larger the group of people, the more likely it is that you're going to have statistical significance with small differences. So obviously this blue dot is a lot different from the other blue dots, but this green dot that we talked about is less different. But if we had more patients, there's a chance that it would become statistically significant. So this is, again, looking at intrahepatic cholangiocarcinoma in the European group of people. And as you can see, it's a smaller group. And so there are no single nucleotide polymorphisms, SNPs, that are rising above that line of statistical significance. And then similarly, this is like looking at an even narrower group. So these are extrahepatic male samples from patients of European descent. And there are, again, no genetic variants that are rising above that line of statistical significance. But in all of these graphs, we are seeing differences. Some of them were above the line of statistical significance. A lot of them were above the line of suspicion. And this gave us enough data to think that an expansion phase was worth it. So Dr. Roberts and Dr. McGlynn were recently awarded an R01 grant by the National Institute of Health. So an R01 grant is the highest level of grant that the government will give you. It's millions of dollars of money to go towards very specific research plan that you set out when you apply. So our goal is to sequence 7,000 patient samples over the next 5 years. So that's, again, to try to increase the sample size so that we're seeing more of the small differences between cases and controls. Currently, we're working with 82 sites from 31 different countries, and that includes the Cholangiocarcinoma Foundation. And I'll talk about that a little more at the end. And something that we're really trying to work towards is emphasizing global genetic diversity and increasing representation in this field of research. And the reason that this is important is that certain genetic variants are more common in certain groups of people. We're all very, very similar genetically, but there are a couple of little things that differ between people of different races and ethnicities. And a lot of these populations are understudied in science. So a lot of research participants up to this point have been overwhelmingly white. It's just a lot of the research has been happening in North America and Europe, so it's partially accessibility and it's partially who is being targeted by researchers. And this is a huge failing with scientific research up to this point. And there are a lot of people who are looking to increase representation in scientific research, and that's a goal of Dr. Roberts. That's something he's really passionate about, is increasing representation in research. And so because of this, we're using this, it's called a chip, called the Global Diversity Array. The specifics aren't as important, but this is looking for over 1.8 million variants, except the baseline for these comes from diverse global populations. A lot of the chips that have been used up until this point, the baseline is coming from overwhelmingly white populations from North America and Europe. So this chip was developed to try to be able to use it in a wider group of people. So it's looking at 4,800 genes and over 1.8 million variants within these genes. And this is the distribution of sites that we currently have. So like I said, we have 82 different sites that are from the countries that are denotated on this map. And something that I want you to recall is the map that I showed at the beginning of the presentation that described the epidemiology of cholangiocarcinoma globally, and thinking about how many countries weren't represented in that, especially looking at all of Africa wasn't represented in that map. And so our goal is really trying to increase the number of centers that we have so that we're getting patients from all sorts of different genetic and lifestyle backgrounds as well. We're not only collecting genetic information through blood samples, but we're also looking to get a lot of clinical information from these patients so that we can look at risk factors that they have and not necessarily just the genetics of their blood. So this is something that is really important to this study. We're still recruiting more sites. Dr. Roberts is a networking fiend. And so he goes to these global meetings and he tries to recruit people from a lot of different sites. Hello. And so you, as cholangiocarcinoma patients and advocates and caregivers, are invited to participate in this GWAS. So we're recruiting both patients and non-patients. It's a fairly simple process. You submit your contact information on the website, and then you will be shipped a blood draw kit. And then you can bring this kit to a clinic of your choice. So maybe your family practice doctor, and they'll do a really quick small volume blood draw, and all costs are going to be reimbursed through our grant. And then blood is shipped to the Mayo Clinic for DNA sequencing. So the QR code on the right side here will bring you to the sign-up sheet. The website is below. And if you go to the Cholangiocarcinoma Foundation's website, under patients on the left-hand side, it says participate in genetic studies. And that's another way to get to this site. And if you have questions about it, you can email Nellie Campbell. She is an amazing resource in our lab, and her email there is below. So we really encourage you to be part of this study and really help us understand genetic differences between people who develop cholangiocarcinoma and people who don't, so that we can work on screening and specific treatments for specific types of cholangiocarcinoma in the future. So these are the people that I want to acknowledge for working really hard on this project. Dr. Roberts' lab, like I said, Nellie is working so hard to ship these kits out and work with all of the sites globally. And then the consortium leadership includes people from all across the United States, from the NCI, Baylor, and this is a list of all of the sites that we're working with globally currently, and we're hoping that the list only gets longer. These are some of the sources that I used. Does anyone have questions?
Perfect. Thank you so much, Caitlin. That was very informative. I will open it up for questions. If you guys want to list your questions in the Q&A box or in the chat box, we will get to them. And I think the first question was, would you please show the initial map you presented?
Yeah.
I was curious too. I was like, I want to see.
Okay, so this was the initial map. This is from a review paper. This isn't one study. It's from a review paper that was trying to compile all epidemiological information on cholangiocarcinoma that has been published. And so looking at the parts of the world that are represented, there's been no published information on cholangiocarcinoma in a majority of countries in the world. And then, so flipping back to the sites that we've currently recruited, we're trying to receive patient samples from as many countries and different global regions as we can. And we're hoping to have enough samples from individual global regions like North America, South America, et cetera, to be able to see see if there are genetic or risk factor differences based on global regions.
Perfect. Will participants receive results from the blood kit submittal?
That is a great question. So when we have this data compiled, we'll be publishing a paper on it. All of the samples are going to be de-identified in the paper and lumped as groups. So kind of like those Manhattan plots that I showed you, that's what's going to be published. So we'll have the final results of this study published, but individual patients won't get their sequencing data back.
Okay. Would you happen to know Fred Hutch in Seattle is involved with your studies?
He is not. Do you want to say or type who he is? I'm going to write down his name.
Well, it is a cancer institute, the Fred Hutch Institute in Seattle.
Oh, okay.
I think maybe she's just asking maybe where she can be involved, but you can go directly to the link on our website and be involved that way. Just you don't really need your doctor to be involved with this. It's something you can do on your own.
Yeah, absolutely. But also, in order to increase the number of samples, it is nice to work with an institution because they can work on recruiting patients themselves. So I will look up, if there's someone that I can contact there. Thank you for that.
Get them involved. Okay. The ATM gene variant runs in our family and was discovered in a family member's testing after diagnosed with cholangiocarcinoma. Is there value in participating in the study to determine risks for other family members?
That is a good question. So I think that's really important to know for your family. like I said, you're not going to get individual sequencing back for those other family members, but knowing that you have that risk factor, it would still be interesting to look at if there are other risk factors or genetic variants that might contribute to who is developing cholangiocarcinoma within your family and who's not. And also, besides benefiting your individual family, participation is going to benefit the cholangiocarcinoma research field and medical practice field in general. So I would really encourage you to participate.
What is the timeline of the next phase?
So we are already receiving samples. We're planning on doing 2 waves of sequencing. So we're trying to get over 2,000 samples in this first wave. And then we're having centers or hospitals recruit individual patients over the course of the next 5 years. So when a patient comes in with cholangiocarcinoma, their doctor will ask them if they're willing to participate in this study, and then they'll be able to be sequenced in the second wave.
Okay. Excuse me. How would you encourage someone to approach their doctors about participating and finding out more information?
Yeah, I see your question. I'm really sorry to hear about your dad. That's a scary place to be right now. Biomarkers in cholangiocarcinoma is a very difficult field, but this genetic testing, hopefully, is going to contribute to that. So I don't think that, you know, like I said, you're not going to receive individual genetic testing back. But if your dad does have markers that led to the, you know, at least partially led to the development of cholangiocarcinoma, that can help the research field and medical practice field in general. And he could talk to his doctor about this specific study, or he could go to the Cholangiocarcinoma Foundation website and get shipped that kit to do a blood draw.
Yes. So I think the thing we should emphasize that this is a research study and not individual results for biomarker testing or genetic testing. Okay. Is there any value or are there any initiatives in specifically studying situations where, for instance, a mother and daughter have cholangiocarcinoma? Would this potentially fast track target areas?
That is an interesting question. That is not something that this study specifically is looking at. The mother and daughter would be treated as, you know, separate patients. The fact that they have both developed cholangiocarcinoma would give us some good information about what genetic variant is causing that. So it still encouraged participation, but I don't know about fast tracking target areas. I don't know of any studies that are looking at the genetics of familial cholangiocarcinoma.
So that the GWAS is not looking at that? Is that what you just said, familial?
No, it's not. That's not information that's currently part of the group of information that we're collecting. It's more about individual patients.
Okay. Again, individual results will not be sent back for anyone who participates. This is for research widely, and we would get the final results when the results are published, correct?
You would get the collected results.
So the question is wondering if I should submit a blood sample for the study for a son who had passed away at 28. He was BRAF. Again, anyone can participate, right?
Yeah, we're looking for both cholangiocarcinoma patients and those unaffected controls that we looked at. So literally anyone you know can participate in this study. The fact that, you know, perhaps the BRF mutation is in your family would be interesting as part of the study.
Perfect. Is this actionable data? Is there a relationship between SNP and VUS? And does this relate at all to pathogenic germline variants?
I think the actionable part of this that we're hoping results from the study is it's not an immediately actionable thing, but we hope to eventually influence clinical practice by identifying genes that might lead to specific monitoring, getting yearly ultrasounds, et cetera, to try to get cholangiocarcinoma diagnosis early in the disease progression. There is a relationship between SNPs and VUSs. So VUS stands for variant of uncertain significance. So VUS is a SNP. There are SNPs that cause disease. There are SNPs that don't cause disease. And when we're looking at the genetic data, there's a lot of SNPs that we don't know whether or not they cause disease. So that's where the VUSs fall on the spectrum. And looking at pathogenic germline variants. So this is germline DNA is like the DNA that your parents pass to you and you would pass to your children, and it does. So in this study, we're not looking at tumor genetics, we're looking at whole person genetics. So we're getting, because tumor genetics can be different than germline genetics. And so we're getting blood samples, so it's not tumor specific.
So that answers the next question. Can you participate even after you've undergone surgery to remove the tumor?
Absolutely, yeah.
Is there a way for you to access DNA of recently deceased cholangiocarcinoma patients from sources like Foundation One to include more data points in your study?
Interesting. I'm not familiar with Foundation One. I will write it down though. I know that there are a lot of organizations that have biobanks. So the Mayo Clinic, for example, when a patient comes in with cholangiocarcinoma and a lot of different types of cancers, and they're consented into a research study in general, their blood will be stored, and then regardless of whether or not they survive, their blood is accessible for research. So depending on the institution, yes, I will look into Foundation One specifically.
Okay. Yeah, I think what they're alluding to is many of our patients have the biomarker testing done, and also a lot of them are wondering if there is tissue or some, form of DNA that is left from a deceased one, if they could use that to participate.
Mm-hmm.
Okay, this one, you might have to help me with this one. For example, there is a relationship between MUTYH and the SNPs, which in the monoallelic case is non-pathogenic. I think that is an add-on from the previous question.
Yeah. I'm not familiar what MUTYH is. Is that a specific gene?
Yeah, I wouldn't know either.
Let's see if James
maybe if he responds back. Okay.
The monoallelic, that is, so since you have two chromosomes, you have two copies of every gene. And sometimes if there's a mutation in one of them, the second copy is enough to keep the protein functioning normally. So I think that this question in general is relating to if there's a SNP in one of the copies, it can still be non-pathogenic, but if there's a SNP in two of the copies, that might be when we see disease.
Got you.
Yeah. Yes, it's a gene for DNA base repair.
Perfect. And thank you. You're welcome. I'm glad that the biomarker testing helped. Okay, when do you expect the results of the study to be published and where?
So like I said, we're doing 2 waves of genetic testing. The first wave will be completed in 2023. There are probably going to be 2 publications that come from that. One is probably going to be specific to the genetics, and then one is probably going to be focusing more on the clinical variables that we're looking at. So hopefully those 2 publications are completed and published within the next 2 years. It's a 5-year study, so the second round of publications are going to be in 2028, 2029. As far as where they're going to be published, I'm not sure yet. There are a lot of scientific journals, and we're going to try to publish them in the one that has the widest readership that we get accepted in.
Perfect. And I just realized that I just read this question to myself and said, you're welcome, and nobody else knew what I was talking about. So to the person who wrote, thank you to CCF for leading them to biomarker testing, that's what I was saying you're welcome about. Okay. And there is another question on timeline. And again, would they have to be blood relatives of the deceased to participate in this study? No.
Nope, anyone.
Okay. Okay, and once again, can you tell us the easiest way to find the page to enroll? Currently not able to write anything down. Yeah,
So,
can we scan the QR code?
Yeah, if you have a smartphone, you can scan the QR code. Otherwise, if you can remember, you go to the Cholangiocarcinoma Foundation website. There are a lot of different tabs across the top. One of them is patients. So if you hover over patients, it pops down with a lot of different options. And on the left-hand side, there's one of the options is participate in a genetic study. So click on that and it'll take you to the information.
Or feel free to reach out to advocacy@curecca.org and we would be happy to send you the link. And I think Emma put the link in the chat here on the web page too, or on the webinar here. So feel free to grab it from there. So what is the reason they are not giving results back to participants? Can you use your large grant to get the help people so that they understand?
Yeah, that is a great question. I think that part of the issue is that it's really hard to understand, not for the patients, but for whatever provider is given the information. It's really hard to understand the results of this. Like I said, there are 1.8 million different variants that are being studied in this, and being marked as being positive for a variant in a certain place is not something that is meaningful in and of itself. It's not like there's like one variant that would be helpful for someone to know about. Each individual variant out of the 1.8 million would have to be investigated by a genetic counselor or a medical geneticist. And the value of the large amounts of data is really in looking at the cases versus the controls. And so ideally, this study is going to provide a panel of genes that are associated with cholangiocarcinoma, and then individual patients would be able to get sequenced for that panel of genes.
Gotcha. And I was just going to say, we will have more information. There are some other studies that are being done with different grants from Cancer Moonshot stuff that I think they're doing more of results back to patients on that genetic aspect. So we can reach out to us. We'll give you that information, but we'd love to see you participate in all of them because overall it's going to help the cholangiocarcinoma community widely and further research for so many things, earlier detection, like you talked about, risk factors, all of that. How significant is the VAF % variant allele frequency when detected in NGS?
Okay, so for everyone, NGS is next generation sequencing. That's what we're looking at in this study. Variant allele frequency. I am trying to think of what specifically that refers to. This is a me not knowing it, not you not knowing what you're talking about. If you can drop it in the chat, maybe I can come back to this.
Okay, got that. How soon must the blood sample be returned to be included in wave 1?
Yeah, that's a good question. We don't have a scheduled date for when we're sending the first wave. The idea is that by mid-2023, we're sending those off to the National Cancer Institute to be sampled or to be sequenced. So
The sooner the better.
Yeah, the sooner the better, within the next 3 months for sure.
Okay. If you have a child that is a minor, can they participate?
Unfortunately, I don't believe so. I think this is 18 years old and above.
Okay. And will there be someone at the annual conference collecting samples? We are working on that. Yes.
Great question.
We're hoping to have everything set up so that, yes, samples can be collected at the annual conference for your participation.
There are emails going around about that today.
Awesome. Okay, let me check the chat for a few more questions. I felt like I had one on Facebook, but I just lost it. Hold 1 second. Yep, I think we got that. We already got that one. Is the same one that came in here. Perfect. I think that is all. So if there are no other questions, we appreciate everyone scanning the QR code, signing up and participating so we can help with the research. Thank you so much, Caitlin, for doing the webinar today and informing us on the research that's being done. We appreciate you being here and teaching us all so much. Have a wonderful day.
Thank you for inviting me.
Yeah,
I really appreciate all you.
Yeah, we appreciate you so much. Thanks, everyone.